Okay, so welcome back to this next video in which we are discussing voltage-gated ion channels. Okay, so we're currently in the process of discussing calcium-gated potassium channels. Okay, and we've just been discussing the big conductance calcium-gated potassium channels. Okay, so what we're now going to move on to is the other types of calcium-gated potassium channel that I said we were going to study in more detail. So remember, all calcium-gated potassium channels are tetramers of alpha subunits of calcium-gated potassium channels. And we've discussed that there are these 8 different genes for alpha subunits of calcium-gated potassium channels that are organized into these 5 different families here, KCA1 through KCA5. I've said chuck away the KCA4 and KCA5 family. Very little's known about them. They're not calcium-gated. Okay, so chuck those away. We're going to study KCA1, KCA2, and KCA3. Okay, and we now have discussed KCA1. We've discussed that KCA1 contains this one gene, KCA1.1, which codes for the alpha subunit of a calcium-gated potassium channel. You can tetramerize 4 of these identical KCA1.1 alpha subunits together to make a big conductance calcium-gated potassium channel. And we discussed that not only are these things gated by calcium, but also, nicely, they're gated by voltage as well. And we've seen how similar their structure is to the voltage-gated potassium channels and how we believe that actually the mechanism by which voltage causes them to open is exactly the same, basically, as for voltage-gated potassium channels. Okay, we also think that the mechanism of permeation and selectivity is the same, and we have analogous types of inactivation as we did in the voltage-gated potassium channels as well, such as that N-type inactivation, albeit by the beta subunits of the BK channel rather than the alpha subunits themselves. Okay, and we also have some form of C-type inactivation, although it's not very well investigated at all at the moment. Okay, right, and then finally we looked at the actual mechanism by which calcium binding to those calcium balls of the RCK2 domains can lead to the actual opening of the V-gate of the BK channel as well. What we now want to turn our attention onto is SK and IK channels. Okay, so I'll remind you that SK channels, standing for small conductance calcium-gated potassium channels, these are channels which are constructed out of 4 alpha subunits that have been made from the KCA2 family genes, which contains 3 different genes, KCA2.1, KCA2.2, and KCA2.3. You can either make homotetramers, where the 4 alpha subunits that you tetramerize together are made from the same gene, basically, or you can make heterotetramers, where you have alpha subunits from different KCA2 genes, basically. Then I'll remind you of the IK channels as well. So IK channels, they are homotetramers of KCA 3.1 type alpha subunits, basically. Okay, now SK channels and IK channels are actually extremely similar. The alpha subunits have the same overall structure, okay, and the gating mechanisms are believed to be very similar. And indeed, there is a movement to having the IK channel here considered another type of SK channel. Okay, right. So what I now want to do then is discuss the structure of the alpha subunits of IK and SK channels. Okay, discuss the gating mechanism by which calcium can open them. Discuss why it is that voltage can't actually open them. Well, we can't really discuss that much, but we'll discuss that voltage doesn't actually open them, and we'll discuss that that's still a mystery as to why, and then we'll discuss their permeation and selectivity, and I'll say a few words about their inactivation and some toxins which block them. Okay, right. So firstly, let me just copy out the important information. So SK channels. We know that the alpha subunits of SK channels are coded for by these important genes, KCA 2.1, KCA 2.2, and then all the way up to KCA 2.3. Okay, so those are the genes coding for the alpha subunits of SK channels here. Okay. Meanwhile, we know that the intermediate conductance calcium-gated potassium channels, the IK channels, they are homotetramers of KCA 3.1 genes, or KCA 3.1 type alpha subunits. Okay, right. So now what I want to discuss then is what is the structure of the alpha subunits that are produced from these 4 genes. So from KCA 2.1, KCA 2.2, KCA 2.3, and KCA 3.1. What do the alpha subunits actually look like? And then I want to discuss how they tetramerize together to make the full tetramer and how they're arranged within the tetramer. Okay, so the first thing I want to just say is that these genes all have other names, okay? And the other names you will often see if you read the literature, so I just want to give you the other names for them. So KCA 2.1 is also called SK1 for obvious reasons, okay? KCA 2.2 is then called SK2, and KCA 2.3 is then called SK3. Okay, so these 3 genes are also called SK1 to SK3, basically. Okay, you might now be thinking, well, is this one called IK1? Okay, well, unfortunately it's not. Okay, instead it's called SK4, reflecting the fact that there is this movement to try and have this one considered a small conductance calcium-gated potassium channel as well, rather than being in a separate class on its own. Okay, right, so be aware of those other names because you will often see them. Okay, right, so now let me show you the structure of an alpha subunit that has been produced from one of these 4 genes, basically. Okay, so I'll start off by drawing the cell membrane here. Okay, and once again, above will represent the extracellular fluid side of this membrane, and below will represent the cytoplasmic side of this membrane. Now, the membrane spanning topology of these alpha subunits, coded for by these 4 genes, is basically exactly the same as the membrane spanning topology of the alpha subunits of voltage-gated potassium channels. Okay, and indeed, the arrangement of the transmembrane domains in the tetramer is believed to be exactly as it is for the alpha subunits of voltage-gated potassium channels. Okay, so let me draw this out. Now, the first important difference then from the alpha subunits of voltage-gated potassium channels is that there is no T1 tetramerization domain here. So that bit's gone. Okay, but here is the amino terminus. Then we've got the first membrane spanning alpha helix, S1, then the second membrane spanning alpha helix, S2, the third membrane spanning alpha helix, S3, fourth, S4, S5, the P-loop, S6, okay, and now something new, okay, a domain that wasn't present in the alpha subunits of voltage-gated potassium channels, okay, which I'll put here. Okay, now this domain is the domain that's going to be extremely important for the calcium gating of these channels, basically, that we're constructing out of these alpha subunits. Okay, so the SK channels and the IK channels. And of course, the voltage-gated potassium channels are not calcium-gated, so their alpha subunits don't need and don't have this domain. Okay, so what's this bright green domain now called? Okay, well, it's called the Calmodulin binding domain. Okay, and for short, the Calmodulin binding domain is often abbreviated to the CAMBD, like so. Okay, so whenever you see CAM written like this, where you've got capital C, lowercase a, capital M, that is the standard way that people denote Calmodulin. So it's a very standard abbreviation for Calmodulin. Okay, and then BD is then just for binding domain, okay, and we will see the function of this domain later on. We'll see that it binds apocalmodulin even before calcium has gone up in the cytoplasm, okay, and how then when calcium does go up in the cytoplasm, that causes the apocalmodulin to turn into a calcium calmodulin complex, and then it leads to the opening of the SK or IK calcium-gated potassium channel, but we'll come on to that in much more detail. Okay, so with these 2 little exceptions on the amino side and the carboxy side, these are basically very similar to the voltage-gated potassium channels. The transmembrane domain is exactly the same, basically. We've lost the T1 tetramerization domain and we've gained this Calmodulin binding domain. Okay, now, let me turn my attention onto then how these transmembrane domains of these alpha subunits of these SK and IK channels are actually arranged in the tetramer. Okay, so this is the structure of a single monomer, okay, a single subunit of one of these IK or SK channels. Okay, but of course they form tetramers, and basically the nice thing here is that we believe that the arrangement of the transmembrane domains within the tetramer is exactly as it is in the voltage-gated potassium channels. Okay, so imagine color-coding all of the bits in exactly like we color-coded them in here, and this is basically the arrangement into a tetramer. Okay, so exactly the same basically. Okay, now we have lost the T1 tetramerization domains, so in this picture you can imagine taking those T1 tetramerization domains out, okay, and then instead adding in these Calmodulin binding domains, which will be dangling underneath the pore-forming domains of each of the individual subunits rather than the voltage sensing domains, okay, because remember the T1 tetramerization domains were at the amino terminus, so they were prior to S1, and therefore they sat underneath their subunits voltage sensing domain, whereas these Calmodulin binding domains are after the S6 membrane spanning alpha helix, so they'll be sitting underneath their pore forming domain, or rather the pore forming domain of their subunit basically. Okay, so that's a change that you'd see on this picture here. Okay, right, so the basic message here is that these things, their structure is extremely similar to what we have already seen with the voltage-gated potassium channels. So that's very nice. Okay, so now let me talk about how you actually get these things to open then. So gating. Okay, so first thing we need to discuss then is why is the channel normally closed? Okay, well again we believe the reason that the channel is normally closed before you have received activation to open via a calcium signal from the cytoplasm is because the S6 helices are impeding on the internal aspect of the pore. So again we believe that you have the same gate consisting of the S6 helices either being inwards, okay, impeding on the internal aspect of the pore and closing the pore by stopping potassium ions from moving through that internal aspect of the pore, or being open where they're pulled back and then you've got a nice wide internal pore. Okay, right, so we believe that the normal gate, the main gate of the channel, again is these S6 helices either being too narrow towards the internal aspect or being pulled back basically to open it up. Okay, right. Now, before I actually talk then about calcium gating these SK and IK channels and how calcium can cause them to open, let me firstly talk about why these things are not voltage gated. Okay, or really what I'm about to just talk about is the fact that they are not voltage gated. Okay, so I'm going to put that in big letters, not voltage gated. Okay, so you cannot get these SK and IK channels to open by depolarizing the electrical potential difference across the cell membrane, and it's still a bit of a mystery as to why that is. Okay, because we've discussed that these things are incredibly analogous in structure to the voltage gated potassium channels. They have all of the same features. Okay, so they have these voltage sensing domains connected to the pore forming domains by S4, S5 linkers. Okay, and you might think, well, maybe there's a really simple explanation for this. For instance, a really simple explanation that you could dream up for why the SK and the IK channels are not voltage gated is maybe quite simply the S4 helices do not have those positively charged residues in. Okay, maybe they've lost some of the arginines and lysines in the S4 helix. Okay, then those helices would no longer respond to the depolarization. You'd no longer get the movement of the voltage sensing domains outwards in response to depolarization, hence you'd no longer get the movement of the S4, S5 linkers, and therefore you'd no longer get the S6 helices being pulled outwards to open the gate. That would explain why these channels are not voltage gated. However, it's not the case. And the S4 helices of the alpha subunits of SK channels and IK channels do have a positive charge. Okay, so everything seems set for these channels to actually be voltage gated, but they're not, basically, and it's still a mystery as to why that is. Okay, but they are not voltage gated. Okay, right, so I can't really take that explanation, I'm afraid, any further because the explanation just doesn't exist at present. Okay, right, but they are not voltage gated, so I might put an exclamation mark there. Okay, right, so let's talk about how they are gated, and they're gated by intracellular calcium. Okay, so when the cytoplasmic calcium concentration goes up, that is going to cause these things to open. Okay, and why might cytoplasmic calcium go up? Well, maybe you've opened a voltage-gated calcium channel in the cell membrane, and that will allow calcium to come into the cell. So remember, calcium concentration in the cytoplasm is usually ridiculously low. It's usually at around 100 nanomolar. Okay, and that's because calcium is actually quite toxic to the cell. Okay, so you don't want it to be chronically at a high level inside the cell, otherwise it will lead to the cell dying. Okay, so normally then, these SK and IK channels, when you've got a cytoplasmic calcium concentration of 100 nanomolar, that's not high enough to activate them. You need to get calcium going up to above that level in order to actually get the SK and IK channels to open. Okay, right, so how do the SK and IK channels then respond to an increase in cytoplasmic calcium? Let's say cytoplasmic calcium has gone up. How are the SK and IK channels going to sense that, and how is that going to lead to the gate opening? We need to know how are we going to move these S6 helices backwards, okay, out of the pore so that they're no longer impeding on ion flow through the internal aspect of the pore. Okay, well, we've seen obviously how this is done in BK channels. Okay, the BK channels, their alpha subunits have those RCK2 domains which have calcium balls which can directly bind calcium. It's different in the case of the alpha subunits of the SK and IK channels. They do not have calcium binding sites of their own. Okay, these domains here, these Calmodulin binding domains, they don't have calcium binding sites of their own. Instead, what it relies on is an intermediate protein of calcium signaling called Calmodulin. So let me tell you about Calmodulin then. Okay, so Calmodulin is a protein that is fantastically important in calcium signaling. Okay, so calcium signaling is all about using rises in intracellular calcium level to change the cell's behavior. Okay, so a huge number of different receptors on the surface of a cell work to change cell behavior by causing calcium level inside the cytoplasm to go transiently up. Okay, now when calcium level goes up in the cytoplasm of the cell, it has a huge number of different effects on a huge number of different proteins, and that's what changes the cell's overall behavior. Okay, however, the huge number of different proteins that you want the calcium signal to affect do not all have binding sites for calcium of their own. Okay, instead nature has a much more efficient way of doing it. Okay, rather than having to build calcium binding sites into all of the different proteins that you want to be affected by calcium, instead what nature has done is it has created an intermediate protein called Calmodulin which can bind calcium ions, change its conformation, then bind to loads of different proteins and affect their behavior. So the calcium signal affects behavior of many proteins, not directly, but instead through Calmodulin. I mean, some proteins are directly influenced by calcium, i.e., for instance, the alpha subunits of BK channels, they directly bind calcium, but many proteins don't directly bind calcium. Instead, they are affected by calcium signals through this intermediate protein called Calmodulin. Okay, so Calmodulin can be in two states basically. Okay, there is what's known as apocalmodulin, okay, which is Calmodulin which doesn't have calcium bound to it. Okay, and I'm now using this abbreviation for Calmodulin, capital C, lowercase a, capital M. Okay, so let me draw you a picture then of apocalmodulin. Okay, so Calmodulin has two lobes, okay? It's often compared to a dumbbell, okay? It has these two lobes, one of which is at the N terminus of the protein and is therefore called the N lobe, okay? And the other which is at the C terminus of the protein and is therefore called the C lobe. Okay, so we have these two lobes of calmodulin, the N lobe and the C lobe. Okay, and these two lobes of calmodulin are connected by a linker here. Okay, which I'll put like so. Okay, now, each of the lobes of calmodulin has a binding, has two binding sites for calcium. Okay, so here are the two calcium binding sites of the N lobe, and here are the two calcium binding sites of the C lobe. So overall, each calmodulin molecule has 4 calcium binding sites, basically. Okay, now, when calcium is at the low level, the 100 nanomolar in the cytoplasm of the cell, all 4 of these calcium binding sites of the calmodulin are empty. Okay? And there's no calcium in any of those 4 binding sites. Okay, and when the calmodulin molecule has no calcium in any of those 4 binding sites, it is in a conformation which is called apocalmodulin. Okay, and in apocalmodulin, it's sort of bent over in this hunched over way, so the two lobes are sort of hunched back towards one another. I've tried to show this linker sort of hunched over a little bit. Okay, so the two lobes are sort of bunched back together, kind of. Okay, and also very importantly, this linker here between the two lobes of the calmodulin is a linear polypeptide. Okay, so it's not got any secondary structure. It's just amino acid after amino acid after amino acid, a beautiful straight polypeptide. Okay, beautifully simple. Okay, so that's the structure of apocalmodulin, or rather, that's my cartoon of the structure of apocalmodulin. Okay, now, when calcium goes up in the cytoplasm, so let's say you get a calcium signal, which we're assuming you do get, what can happen is calcium ions can then bind to the 4 calcium binding sites on the calmodulin molecule, and that then changes the conformation of the calmodulin molecule hugely, and the calmodulin molecule takes on a structure that is now called a calcium calmodulin complex, which for short is abbreviated to Ca2 plus CAN. Okay, and when the calcium is bound to those 4 calcium binding sites, it changes the structure of the calmodulin significantly. Okay, so no longer is the calmodulin going to be sort of bent over like this. So instead what's going to happen is now the two lobes have sort of sprung out away from one another. And in contrast to the whole molecule straightening out, the linker now adopts a much more complicated structure. It adopts a secondary protein structure, specifically an alpha helical structure. Okay, so a spring-like structure. So here now is my alpha helix here. Okay, right, so this is what's going to happen to the calmodulin molecule once it has 4 calciums bound in those 4 calcium binding sites. I'll show these by orange, now smudged black dots. Okay, so here are the 4 calcium ions bound to those 4 calcium binding sites. So we've now got calcium bound in each one of those 4 calcium binding sites. So this now is the structure of a calcium calmodulin complex. And basically what can go and happen now is these calcium calmodulin complexes can now go and interfere with a huge number of different proteins. Okay, and what helps the calcium calmodulin complex to bind to so many different proteins is that this alpha helix here, called the central alpha helix, which I'll colour in purple, is incredibly flexible, and it can actually end up wrapping itself around certain portions of proteins. Okay, so that's really how calcium calmodulin complexes can bind to so many different proteins. Now, usually in calcium signaling, what happens is the apocalmodulin is floating around in the cytoplasm. Okay, then when the calcium signal comes, you get calcium calmodulin complexes being formed, and then the calcium calmodulin complex binds to the protein. However, in the case of the alpha subunits of these IK and SK calcium-gated potassium channels, it works differently. Basically, you get the apocalmodulin binding to this calmodulin binding domain long before there has ever been any calcium signal. So at the moment, cytoplasmic calcium concentration is at 100 nanomolar, very low. The apocalmodulin therefore doesn't have calcium bound to it, and it is in this apocalmodulin state, shown here, okay, hunched over in that way with a linear polypeptide, okay, and these actually combine to the calmodulin binding domain, okay, prior to becoming calcium-calmodulin complexes. That's unusual. Usually they only bind to their targets once they're actually as calcium-calmodulin complexes, okay, but in the case of these alpha subunits of SK and IK channels, the apocalmodulin binds before there is actually a calcium signal. Then, when a calcium signal occurs in the cytoplasm of the cell, what happens is that calcium ions are going to bind to those apocalmodulin proteins that are bound to the alpha subunits of the SK and IK channels. They're going to turn now into calcium-calmodulin complexes, and then that conformational change in the calmodulin molecule will cause a conformational change in the calmodulin binding domains of all four of the alpha subunits of our SK or IK channel, and that will then pull on the C linker here, which links you up to the S6 helix up here, okay, and that is then what's going to pull the S6 helices outwards, okay, so it's a similar idea to the idea that we had underlying the calcium gating mechanism of the BK channels. Again, you've got the domain that is going to actually respond to the calcium linked to the S6 helix by this C linker portion here, okay, and that's how a conformational change in the calcium sensing domain down here can actually lead to the S6 helices being moved outwards, and therefore the opening of the channel. Okay, so that is what is understood of the calcium gating of SK and IK channels, and how you can therefore pull the S6 helices outwards to open the internal aspect of the pore. Okay, right. Then what we need to talk about is permeation and selectivity. Well, that's really simple. It's a potassium channel, so permeation and selectivity is exactly what we've already studied. Okay, again, it's the ascending portion of the P-loop that will be lining the selectivity filter, okay, and that's the only portion of the pore where potassium ions have to give up their hydration shell. So in the internal portion of the pore that is lined by the bottom of S6 here, okay, that portion, once it's open, is wide enough for the potassium ions to come in with their hydration shells. Okay, so only once it wants to go through the selectivity filter does it have to give up its hydration shell, and we know that the reason it can do that is then because it's replacing its hydration shell with those electronegative oxygen atoms that are provided by the residues in the ascending P-loop. Okay, right. So permeation is exactly the same, selectivity is exactly the same again, okay, so potassium ions are just the right size to go through there. They're not too big that they can't fit, and they're not too small that it's not thermodynamically favourable for them to actually give up their hydration shell for those interactions with the electronegative oxygen atoms that line the selectivity filter. Okay, then inactivation. Okay, well, inactivation is really badly understood for the SK and IK channels. Okay, you will notice that these channels, it's nothing to do with voltage changes anymore, it's all to do with calcium. Okay, remember the changes, the inactivation changes that we've discussed before, N-type inactivation and C-type inactivation, those changes were triggered by depolarization. They were gates that occurred that changed to the closed state upon depolarization, and then when you repolarize the electrical potential difference across the cell membrane, they were undone. Okay, they were opened again. Okay, but we're not now changing the electrical potential difference across the cell membrane. This is all about calcium signals now. Okay, so inactivation isn't driven by electrical potential difference changes anymore. Okay, instead it seems to be driven by calcium basically. So if you have a long-term calcium signal, it's not going to actually keep these channels open for all the time that the calcium signal is there. So they do show an inactivation, but it's driven by calcium rather than by voltage. After all, it's calcium now that is driving them to open rather than voltage. Okay, so they show calcium-dependent inactivation rather than voltage-dependent inactivation. Again, very little is understood about that, so I can't really say anything more about inactivation. Okay, right, the last thing then that I want to say about these SK and IK channels is just to talk about a few toxins which block these channels. Okay, so we had charybdotoxin and iberiotoxin, which were scorpion toxins which were capable of binding to the outer aspect of BK channels and blocking the pores so that no potassium can move through it. Okay, there are analogous toxins, indeed, charybdotoxin can block the IK channels, but there's an analogous toxin for the SK channels, okay, which is from bee venom. Okay, so the toxin that can block SK channels from the extracellular aspect, and which is within bee venom, is called apamin. Okay, so that's capable of binding to the outer aspect of SK channels and blocking the pore so that no potassium can move through, even if there is a calcium signal inside the cytoplasm of the cell causing the SK channel to open. Okay, so apamin, a very important experimental drug again. Okay, right. And then charybdotoxin, that scorpion toxin that we saw, which was capable of blocking BK channels, is also capable of blocking IK channels. However, IK channels are insensitive to iberiotoxin. Okay, so that's a way of discerning a difference between a current which is caused by an IK channel and a current which is caused by a BK channel. So if your potassium current across the cell membrane is blocked by charybdotoxin but not by iberiotoxin, then that tells you it's an IK channel that's allowing those potassium ions to cross the cell membrane rather than a BK channel, because if it was a BK channel, iberiotoxin would block it as well as charybdotoxin. Okay, and then you can tell whether it's an SK channel because it will be sensitive to apamin, whereas neither the IK channel or the BK channels are sensitive to apamin. Okay, right, so those are just some toxins to be aware of that are really important experimentally. Okay, right, so that now concludes our discussion of calcium-gated potassium channels. In the next video, what we'll move on to is a really important topic, the voltage-gated sodium channels.